Hello guys, welcome to this YouTube channel. A good day to everyone watching this video. So this is going to be my third video of the biochemistry series where I'm going to start with the introduction to proteins. So it's going to be like kind of basic concepts on proteins that I'm actually going to deal with. Okay, let's get started. So we all know that proteins or peptides, which is like the common name, are nothing but the polymers of amino acids. So we all know that proteins are made up of amino acids, or I would rather say it as amino acids are like the building blocks of proteins. Okay, so amino acids are the building blocks of proteins. Okay, so when two amino acid molecules are covalently joined together through a substituted amide linkage, then there is a formation of what is called a peptide bond. So this peptide bond is a special type of covalent bond which is a substituted amide linkage, and when two amino acid molecules join together to form a peptide bond, it yields a special type of compound called a dipeptide. Okay, because it consists of two amino acids. So such a linkage or the peptide linkage is formed by the removal of the elements of water, which is commonly called the dehydration reaction, from the alpha carboxyl group of one amino acid and the alpha amino group of the second amino acid or the next amino acid. So since it is a dehydration reaction, it is also an example of what is called a condensation reaction. So if you see the picture here that I've taken from the Leninger textbook Principles of Biochemistry, you see that there are two amino acids. This is 1 and this is 2. The R group is immaterial here because it can be anything. It can be any two amino acids. So it is like denoted as R1 and R2. So when these two amino acids come together, the hydroxyl group of the carboxyl group of the first amino acid and the hydrogen from the amino group of the second amino acid are removed in the form of water, and hence you get this special bond called peptide bond, which is CONH bond. Okay, so it is the CONH bond that is called the peptide bond, and this reaction is an example of a dehydration or condensation reaction. So if three amino acids are joined, then you will have two peptide bonds, and the resulting compound is a tripeptide. Similarly, when four amino acids are joined, you get a tetrapeptide and you will have three peptide bonds. And when five amino acids are joined, you get a pentapeptide and you have four peptide bonds. So if you have n amino acids, then you will have n minus 1 peptide bonds. So n amino acids, AA for amino acids, n minus 1 stands for the number of peptide bonds. So it is pretty obvious that for n amino acids you have n minus 1 peptide bonds. So when a few amino acids are joined in this fashion, the structure is called an oligopeptide. Yes. So when only you have few amino acids joining to form a peptide, a sequence of amino acids joining to form a peptide, then if there are only few amino acids, it is called an oligopeptide. However, when you have many amino acids that are being joined, then the product is called a polypeptide. So you should clearly know the difference between what a protein is and what a polypeptide is. So if you see proteins, they have thousands of amino acid residues. Although the terms protein and polypeptide are sometimes used interchangeably, molecules referred to as polypeptides generally have molecular weights below 10,000 Dalton, and those called proteins have higher molecular weights. So you shouldn't be exactly saying that yes, polypeptides are proteins. No, that's sometimes not correct when you talk from a biochemist point of view. So I would recommend you to say that polypeptides are the ones that have lower molecular weights compared to that of proteins. Okay, moving on to the next slide. So, in a peptide, the amino acid residue at the end with the free alpha amino group is called the amino terminal or the N-terminal residue. and the residue at the other end, which has a free carboxyl group, is the carboxyl terminal or the C terminal residue. So when you have a sequence of amino acids that are being linked together to form a peptide, and each amino acid is joined to the other by means of a peptide bond, there will also always be one leftmost amino acid that has a free N terminal or the amino terminal, and there will always be the rightmost amino acid that has a free carboxyl group. Here you have a free amino group, and here you have a free carboxyl group. So this leftmost amino acid that has a free amino terminal is called the amino terminal end or the N terminal end. Okay, so it's called the N terminal end. But if you see the rightmost amino acid that has a free carboxyl group, it's called the carboxy terminal end or the C terminal end. Okay, so you should, in this particular time, you should know what is called the key convention. So when an amino acid sequence of a peptide or a polypeptide or a protein is displayed, the amino terminal end is always placed on the left, whereas the carboxy terminal end is always placed on the right if there is nothing mentioned specifically. So if you have a sequence of amino acids that is being displayed, say in the form of one letter codes like in a question, say in a gate examination, then if there is no other extra information that is being specified, you can definitely observe, you can definitely assume that the leftmost amino acid is going to be the N terminal and the rightmost amino acid is going to be the C terminal. So this is the key convention that is actually followed now. So the sequence is always read from left to right, beginning from the amino terminal end and ending with the C terminal end. So these two points you should remember. Although the hydrolysis of a peptide bond is an exergonic reaction, it occurs only slowly because it has a high activation energy. If you see the formation of a peptide bond, it requires energy, which means it is an endergonic reaction. So if you see formation of a peptide bond, so it is an endergonic reaction. It has some energy that is required to favor its formation. But if you see the hydrolysis or the breakdown of the peptide bond, it is an exergonic reaction, but it only occurs slowly because it has a high activation energy. Why I am stressing on this particular fact is that most of the proteins that we are going to study are made to work in an aqueous biological environment, and as a result, when you are exposing a protein or a peptide in an aqueous environment, it is more susceptible to what is called hydrolysis, that is the attack by water. So even though it is going to be an exergonic reaction favoring its formation, it still doesn't occur fast because it has a high activation energy. So what does this suggest? The peptide bonds in proteins are quite stable with an average half-life, that is T half, of about 7 years under most intracellular conditions. So this half-life and concepts related to this, you'll be learning more in kinetics, or you'll be more familiar if you have good knowledge in kinetics. But all I want you to remember is the peptide bonds in proteins are quite stable because the hydrolysis of the peptide bond, despite being exergonic, it occurs slowly because it has a high activation energy. Okay, proceeding on further. Okay, so peptides, it is a general fact that peptides contain only one free alpha amino group and only one free alpha carboxyl group at opposite ends of the chain, because you can just have two ends for a peptide sequence and one end, the leftmost end, like in through the key convention, it is going to be the N terminal and the rightmost end is going to be the C terminal. Why I am stressing on this fact is that sometimes there can be questions in the gate exam where they give you a sequence of, where they give you a few amino acids in the form of their one letter codes and ask you how many possible combinations are there for, forming a peptide, okay, a polypeptide and things like that. So these groups ionize as they do in free amino acids, of course, because they have a free amino terminal and a free carboxyl terminal. The alpha amino and the alpha carboxyl groups of all non-terminal amino acids are covalently joined in the peptide bonds, and hence they do not ionize and thus do not contribute to the total acid-base behavior of the peptides. So if you see, This is the rest of the peptide, and here I have one peptide bond, right, CONH, and this is the rest of the peptide. So this group, if you see, they are linked by covalent bond, and hence they do not participate in ionization, and hence they do not contribute to the acid-base behavior of the peptides. So the alpha amino and the alpha carboxyl groups of all the non-terminal, these are all non-terminal because they are present somewhere in the middle. So they are non-terminal amino acids. They are covalently joined in the peptide bonds and hence they do not ionize. However, the R groups of some amino acids can ionize, and in a peptide, these contribute to the overall acid-base behavior of the molecules. So if you have amino acids like aspartate or glutamate or histidine or lysine or arginine, these amino acids have polar charged R groups. So in that case, they are going to ionize and they are going to contribute to the overall acid-base behavior of the peptide molecule. Thus, the acid-base behavior of a peptide can be predicted from its free alpha amino terminal, free alpha carboxyl terminal, combined with the nature and the number of its ionizable R groups of the other amino acid residues. Okay. So, like free amino acids, peptides also have a characteristic titration curve and a characteristic isoelectric pH. So, similar to amino acids that has its own isoelectric pH, that is the pi. Even peptides also have their own characteristic titration curve. Like, if you see my previous video, I would have discussed more about the titration curves and things like that. Similarly to amino acids, even peptides also have their characteristic titration curve and also their characteristic isoelectric pH. And at this pH, they do not move in an electric field that is being supplied. So this particular property of peptides is being exploited to separate proteins and peptides in a given sample. So techniques that use this is, one such example is IEF, which is called isoelectric focusing. This is one such example. I will be discussing more about this when I take up the techniques in the future videos. I request all of you to stay tuned. So the pKa value for an ionizable R group can change somewhat when an amino acid becomes a residue in a peptide. So if you study the amino acid separately and it has a, say, ionizable R group, it has one pKa value. However, if you put the amino acid as a residue in a polypeptide, the R group is now in a different environment and hence it has a different pKa value. It can change. So the main reasons for this is the loss of charge in the alpha carboxyl and alpha amino groups, the interactions with other peptide R groups, and other environmental factors that can actually change this pKa value. It doesn't remain the same. It changes when the amino acid is being incorporated into the polypeptide as a residue. Okay. Moving on to the next slide. This is one such simple fact that I want all of you to get it straight away clear, is that no generalizations can be made about the molecular weights of biologically active peptides and proteins in relation to their functions. I can't say this protein it has a high molecular weight and hence it has a lot of functions. So that generalization should not be made. It's totally incorrect. So even the smallest peptides can have biologically important effects. Science says that smaller things or smaller entities should not be ignored. So for example, if you take a number of vertebrate hormones as example, all of them are small peptides. So oxytocin, if you see, it has only 9 amino acid residues and it is secreted by the post pituitary gland and stimulates uterine contractions during the delivery of the baby. And there's one more, the thyrotropin releasing factor, which has only 3 residues, this particular peptide, and it is formed in the hypothalamus, which is an important part of the brain, and it stimulates the release of another hormone, that is thyrotropin, from the anterior pituitary gland. And there are some extremely toxic mushroom poisons such as amanitin. So it's a very toxic peptide, and these are also small peptides, and even many antibiotics are going to be small peptides. Even a recent research has a lot of focus on designing peptide based drugs, which are also again small residue peptides that are aimed to cure various diseases. So some proteins consist of a single polypeptide chain, but others called multi-subunit proteins have 2 or more polypeptides associated non-covalently. So if you take some proteins, they just consist of only one polypeptide chain. But if you take multi-subunit proteins, they are going to have two or more, and they are going to be, and all these chains are going to be associated non-covalently. So the individual polypeptide chains in a multi-subunit protein may be identical or different. Of course, since there are going to be two or more, these chains can either be identical or different. So if at least two are identical, the protein is said to be oligomeric, and the identical units consisting of one or more polypeptides are referred to as protomers. I want these keywords to be well remembered because there can be various assertion reasoning based questions, as well as comprehension based questions, as well as, even if you're lucky, you might even get a recall question. So if at least two are identical, the protein is said to be oligomeric because it's a multi-subunit protein, and the identical units that consist of one or more polypeptide chains are referred to as protomers. So let's take one classical example, that is hemoglobin. I'll be doing one full separate video on hemoglobin, how it functions and how it transports oxygen, the conformational changes, etc. But yeah, for now, I just took the basic facts about hemoglobin as an example. So, for example, it has four polypeptide subunits, of which two are identical alpha chains and two are identical beta chains, and all four are held together by non-covalent interactions. Remember, these are not covalent. So, each alpha subunit is paired in an identical way with a beta subunit within the structure of this multi-subunit protein, so that hemoglobin can be considered either a tetramer, like alpha 2 beta 2, that's the common way you actually write. I mean, many people write hemoglobin as a tetramer because it has 2 alpha and 2 beta, or it can also be called a dimer of alpha beta protomers. Okay, so this alpha beta is regarded as a protomer, and a dimer of it will be a hemoglobin. Okay, yes, moving on. A few proteins, however, can contain two or more polypeptide chains that are linked covalently. So the previous one I talked about non-covalent interactions, but now I'm talking about covalent interactions. For example, if you take the example of insulin, it has two polypeptide chains that are linked by disulfide bonds. In such cases, the individual polypeptides are not considered as subunits but are commonly referred to as simply chains. In that case, you're not supposed to call each polypeptide chain as a subunit, but you call it as simply chains. Okay. So the amino acid composition of proteins is also highly variable. So you can't say that this particular amino acid has to be only this proportion for this particular protein. No, that's not possible. It's going to be highly variable. The 20 common amino acids almost never occur in equal amounts in any protein. So some amino acids may occur only once or not at all in a given type of protein. Others may occur in large numbers, like you just have a repeat of glycine, and then you might not have any histamines, you might not have any glutamines, etc. It can be totally variable. So now, if we want to calculate the approximate number of amino acid residues in a given protein, a simple protein that contains no other chemical constituents, what you have to do is divide the molecular weight of the protein by 110. So I have a protein. I want to know how many amino acid residues are there in this protein. So what do? I divide the molecular weight of this protein by 110. So why 110? I'm going to talk about now. Although if you take all the amino acid residues, like all the amino acids separately, and can take the molecular weight, sum it up and divide it by 20. That is basically when you do the average molecular weight of all the 20 common amino acids, it comes to about 138. Okay, but you cannot divide the molecular weight by 138. Why? Because the smaller amino acids tend to predominate in most proteins. So in this case, you also have to take into account the proportion in which each amino acid actually occurs in a protein. So if we take into account the proportions in which the various amino acids occur in an average protein, that is done by taking a survey. They study like more than 1,000 proteins and then find out how much time glycine occurs and each amino acid occurs, and then they find the proportion. And after finding this proportion, they somewhere incorporate this proportion in the calculation, and they find out that the average molecular weight of protein amino acids, they get the answer to be 128. So it's not 138, it's 128 now. However, When you link two amino acid residues via a peptide bond, I already mentioned that there's going to be removal of water. So what's the molecular weight of water? It's going to be 18. So what do? Is we subtract the molecular weight of water, that is 18, from this 128, and we get the answer to be 110. That's the reason why you have to divide the molecular weight of the protein by 110 in order to get the number of amino acid residues in a given protein. Okay, so this can be a very simple question in GATE, and I request all of you to score a full in this particular question because it's going to be a very good cakewalk. And I have seen few papers asking the same. Okay. So many proteins, like if you take examples of enzymes, what are enzymes? Enzymes are nothing but mostly proteins with catalytic actions. I wouldn't say in all enzymes are proteins, but yeah, most of them are proteins with a catalytic action. They're going to catalyze a given reaction and make them occur at a very good rate. Okay, so many proteins, for example, if you take the enzymes like RNase A and chymotrypsin, I would be discussing the mechanisms by which these enzymes are going to act in my coming lectures. So if you take these two examples, like among many examples, they're going to contain only amino acid residues and no other chemical constituents, and hence they are referred to as simple proteins because they're only going to contain amino acid residues, and there is not going to be any other chemical constituents like ions or things like that. So it's going to be called simple proteins. However, that's not the case with all the proteins. Some proteins contain permanently associated chemical components in addition to these amino acid residues, and hence they are called conjugated proteins. So apart from the amino acid residues, they are also going to contain some extra chemical component, and hence these type of proteins are called conjugated proteins. So the non amino acid part of a conjugated protein is usually called its prosthetic group. So if you take a protein, if it has amino acid, it should have amino acid residues along with the other extra chemical constituents. The chemical constituents are going to be called the prosthetic group. So if you take conjugated protein, it's equal to prosthetic group plus simple protein. That's the basic kind of equation that you can derive. So conjugated proteins are classified on the basis of the chemical nature of their prosthetic groups, of course. So you have different types of conjugated proteins, and they are classified based on what is the nature of their prosthetic group. For example, if you take lipoproteins, they contain lipids as their prosthetic group. If you take glycoproteins, they contain sugar groups. If you take metalloproteins, they contain specific metals. Some proteins may also contain more than one prosthetic group. Usually the prosthetic group here plays a very important role in the protein's biological function. So every protein is designated for a particular biological function, and this prosthetic group actually aids the protein in achieving so. So here is the list of some of the common conjugated proteins that I've actually taken from Leninger's Principle of Biochemistry's biochemistry textbook, and here I would request all of you to kind of know this because they might even ask as a simple match the following question. So if you take lipoproteins, the prosthetic group is going to be lipids. An example is beta-1 lipoprotein of blood. And glycoproteins, you have prosthetic group as carbohydrates, and one such example is immunoglobulin G, that is IgG. I'll be discussing more in the immunology lectures. And you have phosphoproteins that contains phosphate groups as the prosthetic group, and one such example is casein of milk. And you have hemoproteins, and it has heme or the iron porphyrin ring as the prosthetic group, and the example is of course very prominent, that is hemoglobin. And there's going to be flavoproteins, and it contains prosthetic group as flavin nucleotides, and the example is succinate dehydrogenase. It's a very important enzyme in bioenergetics and metabolism. I will be discussing about it in more detail in the coming videos. And you have metalloproteins. So if the prosthetic group is iron, the example is ferritin. If it is zinc, it's alcohol dehydrogenase. If it is calcium, it's calmodulin. If it is molybdenum, it's a dinitrogenase. And if it is copper, it's plastocyanin. So these are some of the examples of the conjugated proteins along with their prosthetic group, and it's pretty easy to remember, so that if a question you get in either say as a match the following question, you should be able to score full. Usually match the followings are asked for like even 2 marks, and it should be a cakewalk. So this is like the end of my first video. So end of my first video on protein concepts. So I just made it as a short video because I want the basics on protein biology to be very clear before I proceed on to other complex topics on protein biology, and hence this video is pretty short because of that. So before I kind of close this video, I would request all of you to buy this updated formula sheet for GATE XL and GATE BT from Miss Priyanka Joshi. It's an updated formula sheet. It is not just the previous formula sheet that is being circulated, and it's just for rupees 99, and it's the offer for all people now. It's not just for the early 20, and you can get in touch with her through the email ID that is provided in the description box of our YouTube channel, as well as through LinkedIn and Instagram IDs that are being provided in the channel. So it is very helpful. Trust me, I used this formula sheet, and I was able to secure a really good rank in my GATE 2023 examination. I would request all the GATE aspirants who are aiming for a really good rank in GATE XL and GATE PT to use this formula sheet at least for your last minute preparation. It is really helpful. So I request all of you to like, share, and subscribe to our channel, and stay tuned for more exciting videos and more content that makes your GATE 2024 preparation really easy. So have a nice day. Thank you and happy learning.